This is Dr. Hutch, and this is a presentation on nervous system part 3, the senses. So the senses have a lot of different functions, one of which is homeostasis, believe it or not, keeping everything in balance, protective mechanisms. And what it does is to provide information about the outside world and then your internal environment as well. And there's two types of senses. General senses, this includes receptors that are widely distributed throughout the body. They're in your skin, various organs, and your joints. And then the special senses, which are specialized receptors confined to the structures in your head. So your eyes, ears, nose, and mouth. Sensory receptors collect information from the environment, then they relay it to the central nervous system on the sensory neurons. They link the nervous system to internal and external changes or events, and they can be specialized cells or multicellular structures. The pathway of sensory information is from the sensory receptors to the central nervous system. So we're talking about the brain or spinal cord. A stimulus first stimulates the sensory receptors, and the stimulus is converted into graded receptor potentials, and that's called transduction. And remember, when we talk about receptor potentials, we're talking about a change in polarity or a change in charge across the membrane. Receptor potentials can trigger action potentials, which are conducted along sensory neurons to the central nervous system, and then this is called transmission. Interpretation occurs in the brain or the spinal cord of the central nervous system. And this figure shows you sensory transduction, transmission, and interpretation from two different sources. The first one is from a free nerve ending, like in the skin, and the second example are receptors like your taste buds. And it goes through the stimulus, transduction, transmission, and then interpretation. Sensory receptors respond to specific stimuli, particularly sensitive to a certain type of environmental change and less sensitive to other stimuli. The receptors allow the body to interpret sensory events. There's 5 types of sensory receptors in the body. Chemoreceptors, which respond to changes in chemical concentrations like smell, taste, and oxygen concentration. Pain receptors, also known as nociceptors, which respond to tissue damage, and mechanical, electrical, thermal energy is usually related to this. Thermoreceptors, which respond to moderate changes in temperature. Mechanoreceptors, which respond to mechanical forces that distort the receptor, like touch, tension, blood pressure, and stretch. And photoreceptors, which respond to light. So we're talking about your eyes. Sensory receptors can take the form of ends of neurons or cells near the extensions of the neurons. Stimulation of the receptor causes local changes in the membrane potential, which causes graded potential according to the stimulus intensity. If a receptor is part of the neuron, the membrane potential can generate an action potential. If the receptor is not part of a neuron, the receptor potential must be transferred to a neuron to trigger an action potential. Peripheral nerves transmit impulses to the central nervous system, where they're analyzed and interpreted in the brain. Let's talk about these terms: sensation, perception, and projection. Sensation occurs when action potentials make the brain aware of a sensory event. For example, awareness of pain. Perception occurs when the brain interprets the sensory impulses. An example is realizing that the pain is the result of stepping on a tack. And projection is a process in which the cerebral cortex interprets sensation as being derived from certain receptors. The brain projects a sensation back to the apparent source, and it allows a person to locate the region of the stimulation. And here we show information flow in the special senses, talking about smell, taste, sight, and hearing. So if we're talking about smell, you have the olfactory receptor cells that are your sensory receptors. If we're talking about taste, it's the taste bud receptors. For sight, it's the rods and cones in the retina. And for hearing, it's the hair cells in the cochlea. Next, impulse in the sensory fibers. In smell, we're talking about olfactory nerve fibers. In taste, sensory fibers in various cranial nerves. Sight, optic nerve fibers. And hearing, auditory nerve fibers. Okay, then the impulses reach the central nervous system. For smell, we're talking about the cerebral cortex. For taste, the cerebral cortex. For sight, midbrain and cerebral cortex. And for hearing, the midbrain and cerebral cortex. For the sensation, for smell, it would be like a pleasant smell. Taste, a sweet taste. Sight, a small round red object. Hearing, a crunching sound. And then finally, perception. Smell, the smell of an apple. Taste, the taste of an apple. Sight, the sight of an apple. And hearing, the sound of biting into an apple. Sensory adaptation is the ability to ignore unimportant or continuous stimuli. It involves a decreased response to a particular stimulus from the receptors, or peripheral adaptation, or along the central nervous system pathways leading to the cerebral cortex. This is central adaptation. When sensory adaptation occurs, sensory impulses become less frequent and they may cease. A stronger stimulus is then required to trigger impulses. This is best accomplished by the thermoreceptors and the olfactory receptors. Your general senses include senses with small, widespread sensory receptors associated with the skin, muscles, joints, and viscera, or your organs. General senses are divided into three groups. The exteroceptive senses. These are senses associated with the body surfaces. Examples include touch, pressure, temperature, and pain. Interoceptive or visceroceptive senses are senses associated with changes in the viscera. Examples, blood pressure, stretching blood vessels. Also, proprioceptive senses. These are senses associated with changes in the muscles, tendons, and joints, and body position. Examples include those that are stimulated when changing position or exercising. There's three types of mechanoreceptors which respond to touch and pressure. Free nerve endings. These are common in the epithelial tissues, and they're the simplest receptors. Examples, you sense itching and other sensations. The tactile Meissner's corpuscles. These are abundant in hairless portions of the skin and lips, and they detect fine touch and texture. Can also distinguish between 2 points. And then the lamellated Pacinian corpuscles. These are nerve endings encased in large ellipsoidal structures. They're common in the deeper subcutaneous tissues and tendons and ligaments. They detect heavy pressure and vibrations. This figure shows you the touch and pressure receptors. Here we have basic free nerve endings in the epithelial cells. Here we show the tactile Meissner corpuscles, and here we show the lamellated Pacinian corpuscles. The temperature senses. Temperature receptors are also referred to as thermoreceptors. They are free nerve endings in your skin. And there's two types of thermoreceptors. You have warm receptors that are sensitive to temperatures above 25 degrees Celsius or 77 Fahrenheit. They are unresponsive to temperatures above 45 degrees Celsius or 113 Fahrenheit. Your colder receptors are sensitive to temperatures of between 10 degrees Celsius or 50 degrees Fahrenheit and 20 degrees Celsius or 68 degrees Fahrenheit. The pain receptors respond to temperatures below 10 degrees Celsius, which produce freezing sensations. And they respond to temperatures above 45 degrees Celsius, which produce burning sensations. The pain receptors or nociceptors consist of free nerve endings, and they're widely distributed. The nervous tissue of the brain lacks pain receptors. They're stimulated by tissue damage, chemicals, mechanical forces, or extremes in temperature and oxygen deficiency. They adapt very little, if at all. Okay, visceral pain. So pain receptors are the only receptors in the viscera whose stimulation produces sensations. Pain receptors in the viscera respond differently to stimulation than those of the surface tissues. Visceral pain may feel as if coming from some other part of the body, and this is called referred pain. Examples of referred pain include heart pain, which often feels like it's coming from the left shoulder or medial portion of the left arm. Referred pain results from common nerve pathways in which sensory impulses from the visceral organs and certain areas of the skin synapse with the same neuron in the central nervous system. This figure shows you where referred pain may occur, and you see certain areas have a much broader referred pain than others. For example, kidney pain. This figure shows you referred pain from the heart and the pathways, and it shows how we have a common endpoint from the heart as well as certain areas of the skin. There's two types of axons or fibers that conduct impulses away from pain receptors. Fast pain A delta fibers, also called myelinatida, they conduct impulses rapidly, up to 30 meters per second. Oh, let me go back one step here. Oh, that's a misspelling by the, I knew that wasn't a real word. Fast pain or A delta fibers, they're myelinated, okay? And they conduct impulses rapidly, up to 30 meters per second. They're associated with sharp or acute pain in localized skin areas, and they usually stop as soon as stimulus stops. The slow pain or C fibers, these are unmyelinated. They conduct impulses slowly, up to 2 meters per second, and they're associated with dull or aching chronic pain. They're difficult to localize. Pain is often continuous after the stimulus stops. In the brain, most pain fibers synapse in the reticular formation, and they proceed to the thalamus, the hypothalamus, and the cerebral cortex. The thalamus begins the sensation of pain. The cerebral cortex judges the intensity of pain, locates a source of pain, and produces emotional and motor responses to pain. The emotional response to pain involves the limbic system. Gray matter in the brainstem regulates the flow of impulses from the spinal cord, and pain-inhibiting substances are produced in the body. These include enkephalins, serotonin, and endorphins. A clinical application. In treating pain, about 25% of people have moderate or severe pain at any time. Treatments for pain include non-steroidal anti-inflammatory drugs, or NSAIDs, such as aspirin, ibuprofen. These can cause GI irritation, however. Opiates can be addicting. Acetaminophen is non-addicting, and there's no GI irritation, but it can damage the liver. Reformulation efforts are producing smaller particles that dissolve faster and relieve pain faster. Chronic pain treatments include NSAIDs, exercises, injection of anesthetics into cramping muscles, antidepressants, transcutaneous electrical nerve stimulation, and invasive nerve block. This shows where we get some of our painkillers from nature, like the willow tree, poppy, and certain shells. Proprioception. So proprioceptors are mechanoreceptors that send information to the central nervous system about body position and length and tension of skeletal muscles. The main types of proprioceptors include lamellated or Pacinian corpuscles. These are pressure receptors in joints, and muscle spindles, which are stretch receptors in skeletal muscles. They initiate stretch reflexes in which spindles stretch causes muscle contraction. Golgi tendon organs. Stretch receptors and tendons. They stimulate reflexes that oppose stretch reflexes. They help maintain posture, and they protect muscle attachments from being pulled loose. This figure shows the stretch receptors. So muscle spindle and Golgi tendon organs are shown here. The visceral senses. These have receptors in internal organs. Examples include lamellated corpuscles, which are free nerve endings also, lamellated corpuscles and free nerve endings. They convey information that includes the senses of fullness after eating a meal, as well as the discomfort of intestinal gas and the pain that signals a heart attack. Table 12.2 goes over the different receptors associated with general senses. Special senses. These are senses that have sensory receptors that are within large, complex sensory organs in the head. Types of special senses in the organs include smell, this is olfactory organs in nasal cavity, taste buds in the oral cavity, hearing and equilibrium in the ears, and sight, which involves the eyes. Clinical application. So there are mixed up senses or synesthesia in certain individuals. This is a condition in which the brain interprets a stimulus from one sense as coming from another. An example, the paint smelled blue. 1 in 2,000 people have this condition, and it lasts a lifetime. It's common in creative people, and is caused by genetic mutation. There's 4 genes that have been identified for this. The most common form is grapheme color type synesthesia, in which letters or numbers or time evoke certain colors. In lexical gustatory synesthesia, this is a name that evokes perception of strong taste or smell. Let's talk about olfaction. This is the sense of smell. Olfactory receptors are olfactory receptor cells that are chemoreceptors, and they respond to chemicals dissolved in liquids. A sense of smell provides 75 to 80% of the sense of taste. Olfactory organs contain olfactory receptor cells, which are bipolar neurons, and supporting epithelial cells. Olfactory neurons have knobs at the distal ends of their dendrites covered with cilia. They cover upper parts of the nasal cavity, superior nasal concha, and a portion of the nasal septum. Odorants may bind to any of almost 400 types of olfactory membrane receptors, resulting in depolarization and action potentials. This figure shows you the olfactory receptors. Once olfactory receptors are stimulated, the nerve impulses travel through the openings in the cribriform plates of the ethmoid bone. It goes from the olfactory nerves to the olfactory bulbs to the olfactory tracts, then to the limbic system for emotions and the olfactory cortex for interpretation. Olfactory bulbs process sensory impulses. The limbic system, which is a center for memory and emotion, provides emotional responses to certain odorant molecules. The leading hypothesis for encoding specific smells is that each olfactory receptor cell contains only one type of membrane protein, and each type of membrane protein can bind several types of odorants. The brain interprets the binding as an olfactory code. The olfactory organs are located high in the nasal cavity above the pathway of the inhaled air. Olfactory receptors undergo sensory adaptation rapidly, and the sense of smell drops by about 50% within 1 second after stimulation. Olfactory receptor neurons are the only ones in direct contact with the environment, and they can be damaged. Receptors are replaced regularly, so this is very unusual for neurons to be replaced in the human adult. Next, let's talk about the sense of taste, or gustation. Gustation is the sense of taste, and taste buds are responsible for this. These are the organs of taste, and they're located in the papillae of the tongue and the roof of the mouth, the linings of your cheeks, as well as the walls of your pharynx. There's about 10,000 taste buds, each with 50 to 150 taste cells. The taste receptors are chemoreceptors. The taste cells are modified epithelial cells that function as receptors. The taste hairs are microvilli that protrude from the taste cells through the pores of the taste buds, and they're sensitive to parts of the taste cells. Taste cells are replaced every 3 days. This figure shows you the papillae with the taste buds, as well as the taste cells and the taste hairs. There's 5 primary taste sensations: sweet, which is stimulated by carbohydrates; sour, which is stimulated by acids; salty, which is stimulated by salts like sodium or potassium; bitter, which is stimulated by many organic compounds like magnesium and calcium salts; and umami, which is stimulated by some amino acids like MSG. Each flavor results from one primary taste sensation or a combination. Spicy foods may stimulate a class of pain receptors. Taste receptors do undergo rapid adaptation. Sensory impulses from taste receptor cells travel in fibers of 3 different cranial nerves according to the location of the taste cells: the facial nerve, the glossopharyngeal nerve, and the vagus nerve. The cranial nerves conduct impulses into the medulla oblongata. Impulses then proceed to the thalamus. Impulses are interpreted in the gustatory cortex and the insula ultimately. There are smell and taste disorders. Disorders of smell and taste can be caused by colds, flu, allergies, nasal polyps, swollen mucous membranes in the nose, head injury, toxic chemical exposure, and nutritional or metabolic problems and diseases. Some cases are temporary and others are permanent. Often the cause cannot be identified. Drugs and medications can also alter your smell or taste. Let's talk about the sense of hearing, your ear. This is the organ of hearing and equilibrium. There's 3 sections of the ear: the outer external ear, the middle ear, and the inner internal ear. Parts of the outer ear include the auricle or pinna, which is a funnel-shaped area which collects sound waves, the external acoustic meatus, which is an S-shaped tube lined with ceruminous glands. It carries the sound to the tympanic membrane and terminates at the tympanic membrane. And then the tympanic membrane, also known as the eardrum, which vibrates in response to sound waves. Parts of the middle ear include the tympanic cavity, which is an air-filled space in the temporal bone, and auditory ossicles, which are 3 tiny bones, which vibrate in response to the tympanic membrane vibrations, which amplify force. And they include the malleus, incus, and stapes, otherwise known as the hammer, anvil, and stirrup. Then you have the oval window. This is an opening in the wall of the tympanic cavity. The stapes vibrates against it to move the fluids in the inner ear. This figure shows you the major parts of the ear, and you do need to get to know these. Here you see the incus, malleus, and stapes. Here's the semicircular canals. Here's the facial nerve, the vestibulocochlear nerve. Here we have the cochlea, the oval window, the round window, and the tympanic membrane. And here you see the auditory tube, and of course the external acoustic meatus. This shows you the relative size of the auditory ossicles compared to a penny. Let's talk about the middle ear and the tympanic reflex. So muscle contractions that occur during loud sounds to lessen the transfer of sound vibrations to the inner ear and prevent damage to the hearing receptors. Muscles involved in this reflex include the tensor tympani, which pulls the malleus away from the tympanic membrane, and the stapedius, which pulls the stapes away from the oval window. This figure shows you a close-up of the tympanic reflex and the auditory ossicles. The auditory or eustachian tube connects the middle ear to the throat. It opens into the nasopharynx of the throat. It helps maintain equal air pressure on both sides of the tympanic membrane. It's usually closed by valve-like flaps in the throat. Changes in altitude may function noticeable. For example, rapid descent in altitude after mountain climbing or during an airplane flight temporarily impairs hearing, as the excess atmospheric pressure causes inward bulging of the tympanic membrane. When the pressure difference reaches a certain point, air travels through the auditory tube to the middle ear, equalizing the pressure and restoring hearing. This is accompanied by a popping sound. Okay, the clinical application in middle ear infections. This is called otitis media. It occurs because of mucous membranes of the auditory tube and middle ear being continuous. Bacteria can travel from the throat or the nasal passages to the middle ear. Greater incidents occur in younger children due to auditory tubes being very short. It's treated with antibiotics. Recurring cases are treated with tympanostomy tubes. which allowed the drainage through small openings in the tympanic membrane. The inner ear is a complex system of labyrinths. The osseous bony labyrinth, which is a bony canal and temporal bone, and it's filled with fluid called perilymph. Then you have the membranous labyrinth. It's a tube of similar shape that lies within the osseous labyrinth. It's filled with fluid called endolymph. And there's three portions of the labyrinths: the cochlea, which functions in hearing, the semicircular canals, which function in dynamic equilibrium, and the vestibule, which functions in static equilibrium. This figure shows you the parts of the internal ears. There are two membrane-covered windows in the wall of the bony labyrinth. The oval window, which is an opening in the wall of the tympanic cavity through which the stapes transfers vibrations to the fluid of the inner ear. Vibrations stimulate the hearing receptors. Then you have the round window. This is a window in the wall of the inner ear facing the tympanic cavity. It dissipates excess vibrations into the tympanic cavity. The cochlea. This is a spiral, snail-shaped tube, which is widest at its base, and it becomes narrower towards the tip. It's coiled around the bony core, or the modiolus. The spiral lamina is a bony shelf that coils around the cochlea. And there's three compartments of the cochlea: the scala vestibuli, which is the upper compartment and part of the bony labyrinth, which leads from the oval window to the apex of the spiral; the scala tympani. This is the lower compartment, which is part of a bony labyrinth. It extends from the apex of the cochlea to the round window; and the cochlear duct. This is a middle compartment, portion of the membranous labyrinth. It contains receptor organs for hearing, like the spiral organ and the organ of Corti. This figure shows you the cochlear structure and the windows of the inner ear. And this figure shows what the cochlea would look like if it were unwound. The cochlea contains three membranes. The vestibular membrane. It separates the scala vestibuli from the cochlear duct. The basilar membrane. It separates the cochlear duct from the scala tympani. It forms the floor of the cochlear duct. And the tectoral membrane. It extends partially into the cochlear duct and forms a roof of the hearing receptor organ, the spiral organ. This figure shows you the cochlea and the spiral organ. Clinical application: getting a cochlear implant. Hearing aids amplify sound, but cochlear implants directly stimulate the auditory nerve. It enables a person to hear certain sounds. The best time is before age 3 to get one, as the brain is rapidly processing speech and hearing. Okay, let's talk about the spiral organ, or the organ of Corti. The spiral organ is an organ for the sense of hearing. It sits on the upper surface of the basilar membrane and contains hearing receptor cells called hair cells. Hair cells contain stereovilli, or stereocilia. A tectoral membrane passes like a roof over the hair cells, and sound vibrations cause the stereocilia to contract and bend against the tectoral membrane. Different frequencies of vibrations move different parts of basilar membranes. The nerve impulses are generated on the fibers of the cochlear nerve, and the receptor cells in different regions of the cochlear duct detect different frequencies of vibration. This figure shows you the spiral organ. This figure shows you a straightened out view of the cochlea. The auditory pathways start with the cochlear branch of the vestibulocochlear nerve, which goes to the medulla oblongata, then to the midbrain, then to the thalamus, and then to the auditory cortex and the temporal lobe of the cerebrum. This figure shows you the auditory nerve pathway. This table goes through the steps in the generation of sensory impulses, as we have talked about previously. This is a good chart to get to know. So first, the sound waves enter the external acoustic meatus. Then the sound waves cause the tympanic membrane to reproduce the vibration coming from the sound wave source. The auditory ossicles amplify and transfer the vibrations to the end of the stapes. The movement of the stapes at the oval window transfers vibrations to the perilymph and the scala vestibuli. The vibrations then pass through the vestibular membrane and enter the endolymph of the cochlear duct. The different frequencies of vibration in the endolymph move the specific regions. A receptor cell depolarizes and its membrane becomes more permeable to calcium ions. In the presence of calcium ions, the vesicle at the base of the receptor cells release neurotransmitter. The neurotransmitter then stimulates the nearby sensory neurons. The sensory impulses are then conducted along the fibers of the cochlear branch of the vestibulocochlear nerve, and the auditory cortex of the temporal lobe interprets the sensory impulses. Hearing loss. About 8% of people have some degree of hearing loss. And there's two major types of hearing loss. Conductive deafness, in which interference with the conduction of the sound vibrations occurs to the inner ear. 95% of the cases of hearing loss are conductive deafness. It's caused by accumulation of earwax, or hardening of or injury of tympanic membranes, or injury to the auditory ossicles, which is autosclerosis. And the diagnostic tests for this are the Rin test and the Weber test. The sensorineural deafness may occur also, which is a damage to the cochlea, auditory, or vestibulocochlear nerve, or the nerve pathways. It can be caused by long-term exposure to very loud sounds, such as factory noise, loud music, and explosions. It's also caused by central nervous system tumors, brain damage resulting from a stroke, and the use of certain drugs. Okay, equilibrium. The feeling of equilibrium balance is derived from two senses, each with its own sensory organs. Static equilibrium senses the position of the head when the body is not moving. The receptors are found in the vestibule of the inner ear. And dynamic equilibrium senses rotation and movement of the head and body. Receptors are found in the semicircular canals. In static equilibrium, the organ is located in the vestibule, which is a chamber between the cochlea and semicircular canals of the inner ear. The utricle and saccule are expanded chambers of the membranous labyrinth of the vestibule, and each chamber contains a macula. This is an organ of static equilibrium. A patch of hair cells and supporting cells are a part of this. The hair cells are embedded in a gelatinous material, with the otoliths, which are calcium carbonate crystals, are on the surface. Gravity pulls on the gelatinous mass when the head changes position. The otoliths shift position and pull on the gelatinous mass and the cilia of the hair cells. Bending of the hair cells results in generation of nerve impulses in the vestibular branch of the vestibulocochlear nerve. This figure shows you the saccule and utricle, which provide the sense of static equilibrium. This figure shows you static equilibrium and the response of the macula to a change in head position. Okay, dynamic equilibrium. Receptors for dynamic equilibrium are found in three semicircular canals of the inner ear. The three canals sit at right angles to each other, and each canal contains an ampulla, which is a swelling of the membranous labyrinth that communicates with the vestibule. The crista ampullaris is a sensory organ for dynamic equilibrium and contains hair cells and supporting cells. It's located in the ampulla of each semicircular canal, and it consists of hair cells whose hairs extend upward into dome-shaped gelatinous masses called the cupula. Rotation of the head or body bends the cupula and stimulates the hair cells. The nerve impulses are sent over the vestibular branch of the vestibulocochlear nerve. These figures show you dynamic equilibrium and the response of the crista ampullaris to head rotation. Okay, and then let's talk about the sense of sight or vision. The visual receptors are found in the eye. The accessory organs for sense of sight are the upper and lower eyelids, or the palpebrae for protection, the eyelashes for protection, lacrimal apparatus for tear production, and extrinsic eye muscles for eye movement. The eyelids are composed of 4 layers: the skin, which is the thinnest in the body, the muscle, which is the orbicularis oculi, which closes the eyelids. The levator palpebrae superioris muscle also opens it. There's also connective tissue, which contains tarsal glands which secrete oil onto your eyelashes, and then conjunctiva, which are mucous membranes that line the eyelids and cover portions of the eyeball. This figure shows you a sagittal section of the closed eyelid. Okay, the visual accessory organs. The lacrimal apparatus includes the lacrimal gland. It's in the orbit lateral to the eye and secretes tears. The canal oculi. These are 2 ducts that collect tears. The lacrimal sac collects tears from the canal oculi, and it lies in the groove in the lacrimal bone. The nasal lacrimal duct. It collects from the lacrimal sac and empties tears into the nasal cavity. And then the lysozyme, which is an antibacterial component of tears. Figure 12.23 shows you the lacrimal apparatus. There are 6 extrinsic eye muscles for eye movement: the superior rectus, which rotates the eye up and medially; the inferior rectus, which rotates the eye down and medially; the medial rectus, which rotates the eye medially; the lateral rectus, which rotates the eye laterally; the superior oblique, which rotates the eye down and laterally; and the inferior oblique, which rotates the eye up and laterally. This figure shows you the extrinsic eye muscles. The eye itself is a hollow spherical organ of sight. The wall of the eye has 3 layers: the outer fibrous tunic, the middle vascular tunic, and the inner nervous tunic. This figure is a transverse section of the eye and a superior view. The outer fibrous tunic consists of the cornea and the sclera. Portions of the outer tunic include the cornea, and this is the anterior 1/6. It's a transparent window of the eye and helps focus light rays, and it transmits and refracts light. And then the sclera, which is the posterior 5/6. It's white, opaque, and tough. It protects the eye and attaches muscles. It's pierced by the optic nerve and blood vessels. Okay, the middle vascular tunic. It consists of the choroid coat, ciliary body, and the iris. Portions of the middle tunic include the choroid coat, which is the posterior 5/6 and provides a blood supply. It also contains melanocytes and has melanin, which absorbs extra light. The ciliary body, which is the anterior portion, this is the thickest portion and it's pigmented. It forms a ring to hold the lens and changes the lens shape for focusing, and contains the ciliary muscles to move the lens and the radiating folds, which are the processes. And then also the iris. This is anterior to the ciliary body in front of the lens. It's pigmented and it controls the light entering the eye. The anterior cavity of the eye. This is a cavity between the cornea and the lens, and it's filled with a watery fluid we call the aqueous humor. The lens is a transparent biconvex area which lies behind the iris, and it's elastic. It's held in place by suspensory ligaments of the ciliary body, and it helps focus light rays. In accommodation, we have a change in the shape of the lens to view close objects. The lens thickens and becomes more convex when focusing on close objects. The lens thins and becomes flatter when focusing on distant objects. The ciliary muscle relaxes the suspensory ligaments during accommodation. This figure shows you the anterior portion of the eye. This figure shows you the lens and ciliary body in a posterior view. This figure shows you the accommodation reflex. Look at the iris. This controls the amount of light entering the eye. It consists of connective tissue and smooth muscle. It's a colored portion of your eye. The pupil is a window or opening in the center of the iris. Dim light stimulates the radial muscles and pupil dilates in this case. Bright light stimulates circular muscles and the pupil constricts. The amount and distribution of melanin determines your eye color. The aqueous humor is a fluid in the anterior cavity of the eye. It fills both the anterior and posterior chambers of the anterior cavity and circulates through the pupil. It's secreted by epithelium on the inner surface of the ciliary body and provides nutrients and maintains shape of the anterior portion of the eye. It leaves the cavity through the scleral venous sinus. This figure shows you the aqueous humor. This figure shows you the iris and how it opens and closes. The posterior cavity is a space enclosed by the lens, ciliary body, and the retina. It contains vitreous humor, which is a thick gel that holds the retina flat against the choroid coat and helps maintain the shape of the eye. The vitreous body is a vitreous body with collagen fibers. The inner tunic consists of the retina. The retina contains visual receptors called photoreceptors, and it's continuous with the optic nerve in the back of the eye. It ends just behind the margin of the ciliary body towards the front of the eye. The macula lutea is a yellowish spot in the retina. The fovea centralis is the center of the macula lutea and produces the sharpest vision. The optic disc is your blind spot. It contains no photoreceptors, and it's found where the nerve fibers from the retina leave the eye to become the optic nerve. It's composed of several layers. From the innermost layer, there are the photoreceptor cells, the horizontal cells, and the bipolar cells, amacrine cells, and ganglion cells. The photoreceptor cells, bipolar cells, and ganglion cells provide a pathway for the impulses triggered by photoreceptors to reach the optic nerves. The horizontal cells and amacrine cells modify and integrate the impulses. This figure shows you the cell layers of the retina. This figure shows you the central part of the retina. This table goes over the different layers of the eyes. Refraction. This is the bending of light which occurs when light waves pass at an angle between the mediums of different densities. Focusing bends the light so the image falls on the fovea centralis. When the surface between the different refractive media is curved, it forms a lens. The convex lenses, which are similar to the cornea and lens of the eye, cause light waves to converge. Concave lenses cause light waves to diverge. So refraction in the eye. As light enters the eye, refraction occurs as it passes through the convex surfaces of the cornea and lens. 75% occurs in the cornea. The image is focused on the retina, and it's upside down in reverse from left to right. The visual cortex corrects the reversals. This figure shows you how light refraction occurs through glass, like it does through a lens. This figure shows you how the light waves pass through the lens. This figure shows you the image of an object on the retina and how it turns it over. There are refraction disorders. In farsightedness of age or presbyopia, this begins after age 45. The accommodation becomes impossible due to the loss of elasticity of the lens capsule. Reading glasses can provide refracting power. In nearsightedness or myopia, the eyeball is too long. Light focuses in front of the retina, creating blurry images for distant objects. It's corrected by concave lenses. In farsightedness or hyperopia, the eyeball is too short. Light focuses in the back of the retina, creating blurry images for close objects. It's corrected by convex lenses. Astigmatism. This is a defect in the curvature of the corneal lens. Some parts of the image are in focus while other parts are not. Visual distortion can be corrected with glasses. This figure shows you refraction disorders. All right, the photoreceptors. These are modified neurons of the retina that sense light. Rods are long, thin projections and are rod-shaped. They contain light-sensitive pigments called rhodopsin, hundreds of times more sensitive to light than the cones. They provide vision in dim light, and they produce vision without color in shades of gray. They produce outlines of objects. Then you have the cones, which are short, blunt projections, which are cone-shaped. They contain light-sensitive pigments called erythrolabe, chlorolabe, and cyanolabe. They provide vision in bright light and produce sharp images. They produce color vision, and the fovea centralis contains only cones. This figure shows you the difference in rods and cones. Rods and cones contain light-sensitive pigments that decompose upon light absorption. Rhodopsin, which is a visual purple, light-sensitive pigments in the disc of the rods. In the presence of light, they decompose into opsin and retinol. This triggers a complex series of reactions that initiates nerve impulses, and the impulses travel along the optic nerve. Iodopsins. These are light-sensitive pigments in the discs of cones. Each type of cone contains different light-sensitive pigments, and each type of cone is sensitive to different wavelengths. Color is perceived and depends on which types of cones are stimulated. Erythrolabe responds to red light, chlorolabe responds to green light, and cyanolabe responds to blue light. This figure shows you rhodopsin in the discs of the rod membranes. Stereoscopic vision provides perception of distance, depth, height, and width of objects. It's possible because of the distance between your pupils. It results from the formation of two slightly different retinal images from the eyes, and overlapping images from the two eyes combine in visual cortex to form a single three-dimensional image of an object. This figure shows you stereoscopic vision. The visual pathway. This proceeds from the ganglion cells of the retina to the optic nerve, then to the optic chiasma, the optic tracts, the thalamus, the optic radiations, and the visual cortex in the occipital lobe of the cerebrum. A few fibers branch off before reaching the thalamus, and they enter the nuclei for visual reflexes. The medial fibers from each optic nerve cross over to the opposite side in the optic chiasma to help with depth perception. Some fibers travel from the thalamus to the brainstem for visual reflexes and controlling the head and eye movements while tracking objects. This figure shows you the visual pathways. Lifespan changes. Age-related hearing loss can be due to damage to hair cells in the spiral organ, a degeneration of neural pathways to the brain, and tinnitus may occur. Age-related visual problems include dry eyes, floaters, which are crystals in the vitreous humor, loss of elasticity of the lens, decreasing your accommodation, glaucoma, cataracts, and macular degeneration. And there's age-related smell and taste problems due to the loss of olfactory receptors called anosmia. And that's the end of the section on the senses.